Hello, today we complete our final part of our chapter on bacteria, and we're on to our section on food poisoning. Now, at the beginning of this chapter, I did say to you that twice we have been asked about bacteria on long questions. One was in 2013 and the other was in 2019. And apologies there, that should read question 3 on 2013. So just to remove that there. So question 3, 2013. Then 6, and that was on section B. And then in 2019, 3 years later, again on question 3, section B, just to put all of that there, we note that it was asked again. Now, I said to you in a previous lesson, the syllabus says that we have to know 3 food poisoning bacteria, not 4, not 5. So where I have 6 different food poisoning bacteria mentioned here, you do not have to learn all of them. I have chosen 3 for you: Salmonella, E-Coli 0157, and Clostridium botulinum. Now, we've mentioned quite a bit about Clostridium botulinum, but the 2 sample answers that I will be going through on our online grind class are on Salmonella and also on E. coli 0157. So I'm just going to highlight our 2 from here. So Salmonella is 1. So we just get the highlighter out here. So Salmonella, and the second one we're going to look at is E. coli 0157. I would prefer that you learnt the 2 food poisoning bacteria in detail from the sample answers rather than the chapter here. So our other one then is Salmonella. So if we just go and look at our notes here, so what do we know about Salmonella? So just say, for example, we're talking about the requirements for growth. Salmonella happens to be rod-shaped, has no flagellae, so it is a gram-positive bacteria, and we have 5 requirements for growth. Reminder, what is needed for the growth of Salmonella? Now, I'm just doing the framework here. You will learn the full detailed answers because it's 6 years, 2019, since we've last seen it. So first of all, Salmonella need moisture, and they need it in a liquid form. So must be in a liquid form. They cannot utilize ice, so I'll just say no ice. Next, we note they need correct oxygen level, and make sure that you say correct oxygen level, because reminder, we have aerobic, anaerobic, and we have facultative. Now, Salmonella happens to be what we call a facultative anaerobe. So that means that it can adapt to either situation, with or without the presence of oxygen, but if it had its way, Salmonella would much prefer an anaerobic environment. Next, we have correct pH, and you may recall, well, I hope you do, that it liked a slightly acidic pH of 6.5. Of course, no microorganism can grow without food, and Salmonella happens to be both parasitic and saprophytic. But we emphasize the fact that it's a saprophyte and how it grows on dead or decaying matter. And finally then we have warmth. And in terms of temperature, stick to the language used for the temperature range. happens to be a mesophile or mesophile, and that's 20 degrees to 45, but it includes body temperature, which is a temperature that Salmonella considers to be its very optimum temperature. So they are the conditions for growth of Salmonella. Then we note reproduction. If we're asked about reproduction of Salmonella, it's going to be by binary fission. So reproduction, we'd have to mention binary fission. Then two other terms that would have to be mentioned, how it's asexual reproduction and involves cell division caused by mitosis. Then you would draw your diagrams. Remember, it's rod shaped, how it divides again. Just doing it very roughly here. And I'm just going to put beside that C sample answers from 2013 and 2019, question 3B on food poisoning. Now, sometimes students confuse the difference between food sources and also what we call source. So just going to differentiate here. So the source is where it comes from. So when you're talking about the source of Salmonella, two sources. One, soil. Remember I said to you in one lesson, we have to be careful of uncooked or raw cookie dough, because Salmonella can live in soil. Wheat grows in soil, wheat is turned into flour, flour is made into cookie dough, but there's no heat along the way, and therefore the Salmonella could still actually be alive in the raw cookie dough. So we need to be careful of that. Secondly, the intestines, and so we find Salmonella in the feces. So if you're asked the source of one food poisoning bacteria that you've studied, you have chosen to write about Salmonella. One source is soil and the other source is feces, human feces and animal feces. And we note that chickens, birds, their feces can contain Salmonella, and because the fact that birds are reared in maybe an intensive environment where there's a lot of birds together, it can transfer from one bird to another very quickly. Then, as well as source, you could be asked a term called high risk foods. Now, in terms of the number of high risk foods that you would give, I'm going to take 5 of them. So first of all, raw chicken is a most obvious one. Raw cookie dough is another. Raw sausages could be another one, or should we call it raw cookie dough or uncooked cookie dough might be one there. Eggs, raw eggs, and unpasteurized dairy products. Say like cheese. And finally, we've one other word for Salmonella, and it's called incubation period. Now, what exactly is the incubation period? There's a definition in your notes. The incubation period is the time between ingestion of the raw food or the undercooked food, i.e. the time between the ingestion of the live Salmonella into the body and the onset of symptoms. Just to look there at our chapter, on the bottom of the second page here, I'm just going to write learn, and it's incubation period. So it is the time from exposure to the toxin or the live bacteria. So from the live bacteria or toxin, depending on the type of food poisoning, to the onset of the symptoms of food poisoning. And you can see there that the incubation period for Salmonella is between 12 and 36 hours. So our incubation period, 12 to 36 hours. So the minimum time, it'll take at least 12 hours is what we're saying, from the time you ingest the live Salmonella to the symptoms. And I should actually say, apologies there, the symptoms then would need to be learned. So even if you took 4 symptoms, which we also have here in our chapter. So with regard to the symptoms of Salmonella, we have fever, diarrhea, abdominal pain, and vomiting. So I wouldn't learn it from, as I say, that table. So fever, abdominal pain, vomiting, and diarrhea. So just to recap on Salmonella. So in 2013 and 2019, they said take one, remember they can't be specific, take one food poisoning that you have studied, and you've chosen Salmonella. And just to know that the food poisoning caused by Salmonella is called Salmonellosis, and it's infectious food poisoning. So infectious food poisoning. Requirements for growth: water in a liquid form, facultative anaerobe, pH of 6.5, slightly acidic, parasitic or saprophytic. We focus on saprophytic, and they happen to be mesophiles. Excuse me. Right about reproduction, you have to mention binary fission. Words like asexual reproduction and mitosis must be given. Soil and feces are a source. High risk foods, we said to know 5, and incubation period 12 to 36 hours, and no 4 symptoms. Excuse me. And that's our Salmonella. Now our second one, and like that you're going to learn it from the sample answer. So we're taking E-Coli 0157. So requirements for growth. We have 5. Now they're very similar to Salmonella. So moisture in a liquid form. I'm just doing the breakdown or a framework here. No ice. Secondly, we have our pH, and the correct pH, we note that E. coli neutral pH, which is a pH of 7. Then we need food. They happen to be parasitic or saprophytic. We're focusing on being saprophytic. Fourthly, we need correct oxygen level. And what we can say about E. coli 0157 is that it's aerobic. So it must have, or it requires, oxygen to grow or to reproduce. Like a higher level student would be expected to give this level of detail. You can't just say for requirements for growth, food, warmth, moisture, correct pH, correct oxygen level. You have to be specific. And then we'll take one more then, which happens to be warmth. Now, like Salmonella, we note that E. coli happens to be a mesophile or mesophile that's a range between 20 and 45 degrees centigrade, but it includes body temperature of 37 degrees centigrade. So just to recap there, we have moisture in a liquid form. For their metabolism, they cannot use ice. Correct pH, neutral pH of 7. They're parasitic or saprophytic. We looked at mode of nutrition, saprophytic. E. coli 0157 secretes enzymes into food, breaks down the nutrients, etc. They happen to be aerobic in terms of their correct oxygen level, and they're mesophiles regarding warmth. Next, we note the source. Now, source is not food, just so you know that. Source of Salmonella, we had soil and we had feces. Now, for E. coli 0157, one source would be contaminated water. So, you know, something like a water fountain, where the water coming through the water fountain is not clear, and also things like swimming pools, lakes, and so on. Maybe you've got little kiddies out in stagnant water, out in muddy pools. Another one then for would be the intestinal tract of animals. So, and then thirdly then, unwashed hands. Then we have our high risk foods for E. coli. So we'll take our 5 foods there. Now, raw poultry. So that would be raw birds, etc., like raw chicken, unpasteurized milk. Then we have undercooked meat, like beef. Another one then would be, trying to think there now. Unwashed. Say fruit, vegetables, etc. And I have to go back to my chapter here. And so milk, water, and undercooked mincemeat, or say undercooked burgers, which we had earlier. So undercooked beef burgers, meatballs, and that's repetition there now, isn't it? Just stuck in there. So if the food was contaminated with feces, right, our incubation period there, anywhere from 1 to 10 days, and our 5 then, abdominal pain, nausea, cramps, and this is unusual, kidney failure. So abdominal pain, 1, cramps, nausea, diarrhea, and kidney failure. So we'll just take our symptoms there. Of the food poisoning. So it is a matter of sitting down. So nausea, cramps, abdominal pain, kidney failure in severe cases for very vulnerable, say people like children or people with underlying health conditions. which obviously could result in death. And then 1 to 10 days would be the incubation period. Now, I can't say enough about learning off, as I say, the sample answers for the 2 food poisoning bacteria that we've just looked at. So just to recap, 3 food poisoning bacteria: Salmonella, E. coli 0157, and Clostridium botulinum. NB, the 2 that you will learn in detail. Looking at question 3B on 2013-2019 are Salmonella and E. coli 0157. We've gone through the main parts of it there, but I would learn off these samples. Now, just to finish here, we want to look at the advantages and disadvantages of bacteria and fungi. So, importance of bacteria, that would be the merits, or it could also be the advantages of bacteria. Remember, we said at the beginning of chapter there's desirable effects and then there's undesirable effects with bacteria. So the production of yogurt, cheese, with our Lactobacillus bulgaricus in yogurt, Lactobacillus helveticus in cheese. We've already looked at stage 2 of vinegar production, where the alcohol is made by the yeast in the first stage. And then you might have the alcohol cider, red wine, white wine, add acetobacter bacteria to the alcohol to result in what used to be called acetic acid, now known as ethanoic acid, or vinegar to you and I. Also, bacteria, remember from way back when, are produced in our colon. Vitamin B and K are made by these bacteria. So we'll just take those 3. Disadvantages then, the spoilage of food, trimethylamine in fish, or the production of lactic acid in milk to cause souring. Human disease like meningitis or whooping cough. Food poisoning like botulism, listeriosis, salmonellosis, campylobacteriosis. Then fungi, we've not covered mold just yet. In fact, I'll be giving you a very detailed note rather than just what we have here on the role of bacteria, mold and yeast in food production. So the manufacture of cheese, blue-veined cheese, the penicillium mold is used in. So blue-veined cheese like Stilton, Roquefort, Cashel Blue cheese, used to make the antibiotic penicillin, yeast used in brewing, red bread making, and then edible fungi like mushrooms and truffles. Disadvantages, many fungi are poisonous to humans. You probably are aware that you have to be very careful if you're out picking wild mushrooms, that you know exactly what you're foraging for, as some mushrooms are extremely poisonous, like death cap and panther cap. Plant diseases like Dutch elm is caused by a fungus. Mold is a fungus but can cause bread mold or soft rot on vegetables, where say carrots, it's all fluffy on the surface, mold, and then underneath the fluffy mold you have squelch, this liquefied wet soft rot. Or human disease like athlete's foot and ringworm. Now what I would say to you is I would know 3 of each. For the merits, advantages, benefits of bacteria and fungi, and then 3 disadvantages of them. So that completes our chapter. Quite a lot of detail, and I have gone through it quite thoroughly there. Just as I say, I'm very, very cautious of the fact that we've not seen bacteria since 2019, and the fact of that outbreak of botulism. You know, the way I keep saying it's a living subject, and that might have brought food poisoning to the attention of whoever is writing out the exam questions. So I enjoyed that chapter. I have to say I do love microbiology. So that's the end of today's lesson.